let's start. This is the last session of this morning. Interesting. And I'd like to introduce the last talk. We have the pleasure to have here Vamshi Rao. He is a senior medical affairs neurology in Ultragenyx. And he talks about a GTX 102 clinical developmental update. Please welcome.
Hello, everyone. I'm going to talk slow. I've been told I should talk slow, so I'm going to try. If I speed up, please let me know. First of all, thank you so much for Fast Italia for inviting me here. Thank you, Benedetto. This has been a wonderful conference so far. But before anything, I just wanted to take the moment to say Happy International Angelman Day. The very fact that you have an Angelman Day is like a testament to your perseverance, your hard work. So I'm glad you gave yourself an applause, because if you hadn't, I would have applauded you. But I'll be saying thank you a lot in the talk today, because I truly mean it from the bottom of my heart. My career, my background in child neurology has been inspired by communities like you. I was in rare disease on the neuromuscular side, and I am in rare disease at Ultragenyx because of all the stuff that you do to get to be here. And please don't give up. As Allison said, never, never, ever give up. Carry on. So my job today, and I'm really thankful, is to present the update from the phase 1/2 trial for Ultragenyx, but also talk to you about sort of the phase 3 plans from Ultragenyx. So with that, I want to start, again, like I said, I'll be saying many thank yous. So one of the thank you is to all the investigators on this list here who were helpful in doing the phase 1/2. But more importantly, thank you to everybody who participated, the families. It's not easy to do clinical trials. And all of us on the clinical side know that. And we're extremely, extremely thankful because you participated. So before we go further, I have to give you some disclaimers and level set. GTX 102 is an investigational drug. What I'm about to present is open label data. So you have to interpret it with the due amount of caution. We are going to talk about safety and efficacy together, so it needs to be really balanced. So I want to make sure that we go through all of that. And last disclaimer, as was mentioned, I am an employee of Ultragenyx. So who are we? I'm just going to spend a little time here. We are a company that was founded, really our foundation, our basis is rare and ultra-rare disease. And we really honor what you tell us. We really take what you tell us to develop and design things. It's important for us to hear what the community has to say. And it sort of not only helps us to design smarter, but it's innovation that we can bring earlier and faster. So it's really helpful that we continue to talk to you, you continue to talk to us. Okay, so GTX 102, and I'm not going to go into extreme detail because you've heard this yesterday from Allison, you've heard this today from Dr. Barry Kravis, but the genetics of the disease and how GTX 102, just going to repeat a little bit of it, just for, because repetition never hurts. You know that the lack of UB3A, the protein itself, is the reason for Angelman syndrome. And in the deletion cases, you have the maternal copy or mother's copy not being able to produce because it's deleted. And the paternal copy has this phenomena called imprinting on there, where this antisense transcript doesn't allow the normal process of transcription and then resulting in a UB3A protein. So what GTX-102 does, it's like specifically engineered and designed so that it can block this antisense transcript and let the UB3A inherently in the paternal chromosome in the paternal gene to go forward and make the protein. Again, I have to mention it's an investigational product. So our phase 1/2 clinical study leverages this sort of mechanism and the possibility that this mechanism could have clinical improvement and an effect on some of these domains that you have told us are important to you. So we studied cognition, communication, hyperactivity, sleep, and gross motor. And we observed that there was improvement in this, as was shown by Dr. Perry Travis, and I'll go through some of this to get into a little bit more detail. So you are probably very well versed with this by now, and you can probably teach me the whole clinical trial process. It was really beautifully detailed by Allison too. But I just want to make it a little bit more specific to where GTX-102 is in this process. So as you know, there are these phases where you're trying to figure out safety first in human with phase 1. Phase 2, you go into mostly safety, a little bit of efficacy, and then you take that, inform yourself, and then design the phase 3 trial. And then hopefully, partnering with all our regulatory agencies, listening to feedback, we can get to a point where we have approved drugs. So we are done with our phase 1 and 2. We have finished enrollment. There were 74 participants. We are in phase 3, and I'm really, really pleased to tell you that enrollment has started. We have enrolled the first participants in December of 2024. So that's a big, big milestone for the community and for all of us. We are also planning a study that goes beyond what we are studying in phase 1, 2, and phase 3 in terms of genotypes and the ages that we have studied them. So I'll detail that in the next slide a little bit more. And hopefully with all of this, like I said, we're moving forward, onward and forward, and hopefully we have a treatment for all of us and multiple treatments for all of us. So how are we studying this? Like I said, we finished phase 1 and 2, and here it was really trying to get into the human clinical trials, which, as you heard, are really, really difficult initially, but we got there. We completed enrollment, and it has really given us an understanding of the safety and the efficacy of the drug. Before I go to long-term extension, phase 3 is the study that we are in right now, and we are enrolling 4 to 17-year-olds. It's a 2-year study. It's a global study. The enrollment, as I said, has begun. The long-term extension study is something where we are going to have GTX-102 available to all participants who have participated in other GTX-102 studies, so that we can continue to study safety and efficacy, going forward in the long term to make sure that we understand fully and in the long run. And this is another exciting part I wanted to tell you about. We are planning a study called the Aurora study, and this is the study where we are expanding the age. We're going below 4, we are going above 17, but we're also studying other genotypes. So this is sort of our study beyond the phase 3 with expansion. And what I wanted to tell you today is that we are planning to open enrollment in 2025. And it is also a global study. All of this information, the place where the truth resides, is in all these websites. And we continuously try to update us as we get information. So it's the best place to check for where trials are happening and how the sites are opening up, and we keep providing information. So let's look into the phase 1/2 study results. And before we go there, just a little bit of what we are going to be looking at and how it was done. So we're going to look at cohorts 4 to 7 and A and B. And the reason being that these are the cohorts that have the longest term data, but these also are the cohorts, we call them the dose expansion, and dose escalation cohorts. And these are cohorts where we have the longest term data. So it's a good group to sort of watch together. The way the trial was designed was that we had given 4 loading doses, and then we gave maintenance doses every 3 months. And we studied selected endpoints there. Again, the really important endpoints that we've all heard from you as a community, which was cognition, communication, sleep, behavior, and motor function. And obviously, and very importantly, you have to study safety in a trial. And in this this phase, you really have to make sure that there's a safety profile that is well known. So this table, I'll just walk you through it. You don't have to read everything. But a table like this is basically to look at how your participants are matched. So what you're doing is, as your eyes go down column 4 to 7 in A and B, and as you look at the characteristics with age, baseline, scores, and everything, you want to see that they are well matched before you start heading into looking at the data. So as you look here, they are well matched. And the other thing, before we dive into the data, is to know that it's a phase 1/2, and it's an open label study, right? So you don't have a placebo sort of an arm or a comparator arm in the study itself. So you have to use something to compare against. And this is another thanks to you. This is because of the community that we can actually leverage this amazing data, which is natural history data. And it is never possible to look at phase 1/2 data sometimes when you don't have a good natural history data. So thank you so much. It's not easy to do natural history. Again, when you know that you're doing natural history for something in the future, it's not really letting you do a drug at the same time. And you're visiting clinics and collecting. This takes time. So thank you very much. And what we did when we compared the phase 1/2 data is to look at the 4 to 17-year-old with the deletion type within this natural history data. So we're sort of comparing 2 sort of similar things. So let's dive into the data. So one of the first things I want to highlight was we looked at the cognitive scores, and this is by using the Bailey-4. And Dr. Berry Kravis went before and it was wonderful. She set the stage. It is a tough test. Again, thank you. It's a tough test. It's not foolproof, but it is the best we have. And until we start making all the improvements, that's what we used. What you're seeing here are graphs. On the left side is what we call the GSV scores, and on the right side is the raw score. The GSV score takes the raw score and sort of mathematically converts it so that it's more linear, and you can look across time. You can look at time and across population. So it sort of makes it simpler to compare. But we also have to look at the raw score, and we've been asked to look at the raw score as something that is an endpoint. So we've done our due diligence. What I want to show you here is, as you look at the 1-year time point and then day 506, and you look at the GSV and the raw scores, I just want to highlight that they look similar, right? They're both improving, so that's good news. But when you look at the GSV score, you also can use what's in the Bailey manual as a threshold. That dotted line that goes across at 5 is something that the Bailey manual says is statistically significant and not due to random chance, right? So you want to make sure that when you see change, that it's above that. So you're seeing change at year 1, but it not only continues, it actually improves at day 506. So that's really heartening to know that the cognitive domain changes. A lot of those numbers at the bottom, that's probably making you dizzy a little bit, are numbers that are called power calculations. And power calculations is a way in statistics to make sure that you have enough in terms of what you're looking at and in comparison to know that you're powered to study this endpoint, and statistics supports that. And you're not going to see change due to just randomness, it's going to be real. So what this tells us is our power, either way, when you look at it, is above 90%, which makes us confident that we are ready for phase 3 to use this as one of our endpoints, and this is our primary endpoint. We all know Angelman is not one endpoint. And Dr. Berry-Kravis told you can't just look at one endpoint and choose one endpoint, but you have to, because that's what the agencies want you to. But you have to look at the totality of what makes Angelman syndrome and what makes each individual different. So when you look at other domains, one of the domains to look at is receptive communication. And that is one of the domains we looked at. And if you see these two graphs, you're looking on the left side at cohorts 4 to 7, A and B, and the right side, you're looking at natural history. And if you look at what the cohorts that were treated did, they all went above that 6 sort of threshold. And if you look at natural history, it barely increases by a point or so. So this does show that there is an improvement, and it's sustained till the 1-year end mark. Now, one thing that I did not mention in cognition is, what would that mean? And we've heard families saying that cognition could mean that you're engaging, you just seem more alert. When you look at receptive communication, it could mean that you can understand better. Maybe that all leads to then doing your tasks more or being more engaged. So these are definitely some of the implications of this, what families have told us. Now, one thing I want to again level set is, everybody reacts differently in a clinical trial. So you just want to make sure that you realize that everybody can have different changes and to different degree those changes. So it's important to go through all these domains because some may not improve in one domain and may improve in the other domain. And that totality is really important. So when we look at sleep, and Dr. Berry-Kravis mentioned this, we use what are called sleep anchors, which means that we looked at a questions criteria where we scored somebody on a scale from 1 to 7, 1 meaning that they're not at all affected and 7 meaning that they're severely impaired. So a graph on the left with a decreasing score means that you see improvement, because the questions are things like awakening at night, duration of sleep, time to sleep onset, daytime sleepiness, and things like that. So improvement is usually a decrease in the scores, and we saw the improvement in both cohorts 4 to 7 and A and B. Now this could mean not just sleep for the individual themselves, but sleep is something, as all of you know, affects families as a unit. So it's really important domain to consider. It's not just about the kid. It's really something bigger than that. Now, gross motor is a similar sort of scale, again, using the ASA, and ASA is the Angelman Severity Assessment Scale. And the subdomain here is gross motor. And again, you're looking at a scale where a lower score means improvement. And we did see that there was improvement. And gross motor to us also means that, it sort of opens the world up a little bit for the kid. You can walk out without the fear of falling. You don't have to keep that hand in the back, in the small of the back. You guys know that. And you're not worried as much. You can sort of let go of the hand sometimes. So again, a very important domain. ABCC, or the Aberrant Behavior Checklist for community, there's a subdomain called hyperactivity or noncompliance. And we looked at this because we do think that this really impacts, and you've told us that this impacts life. And you see that, again, there's an improvement as detailed on the left side with a score that's lower in terms of hyperactivity and noncompliance. And when you compare that to natural history at 1 year, it kind of doesn't really change much, depending on which natural history you look at. But the change, even if there is, it's very minor with sort of change in age, maybe maturity, but doesn't change too much. So the fact that we saw improvement could mean that you're sort of a little bit more cooperative when you're doing your therapies. and a little less disruptive as the family is going out for dinner or celebrating an event or something like that. This is something that is very dear to our heart, and Dr. Berika has also mentioned this. Angelman is not a monolith. It's not one thing and one thing only. Genetic diseases cannot be that. And so you have to look at multi-domain. And so this is something called the multi-domain responder index. What I'm showing you here are cohorts A and B at the 1-year mark. In the column section, as you look down, you're seeing the different domains that we went through: sleep, motor, communication, cognition, hyperactivity. Each row is a participant. The color represents a certain threshold above which we think that there is a clinically meaningful change. So if that change happened in the study, it's colored green. If there was no change, it's white, and if they worsened, it's pink. So if you look across each participant, you'll notice that certain participants have a change in all domains, and some participants have a change in maybe one domain. And if you take it all together and compare it at that 1-year mark, it looks like at least 79% of participants actually had improvement in at least one domain at year 1. So this tells us that you kind of have to look at sort of the totality of an individual. Another way to look at this is to say, okay, you're changing these domains. What does the change within the domain mean? So what I'm showing you here is cohorts 4 to 7 that was shown in October 2023. But this sort of illustrates, if you look at what changes within domains for certain participants, you can see that it could mean things like following direction, maybe babbling or communicating their wants and needs, to reduced disruptive behavior, better sleep in terms of less awakening, maybe better in opening or closing lids, or maybe throwing or catching a ball. So if you dig deeper, it could mean these are the changes. But again, disclaimer here is not everybody has the changes that are, not everybody has the same changes and the same degree of change. So it's really important to look at it in a very nuanced, finer way. We obviously have to look at safety and continue to look at safety throughout all the trials. But in the phase 1/2 trial, what is listed up there is the most commonly reported events. Now, all the clinicians who were in clinical trials reported that, told you that you have to report everything, and you have to report everything. Then you judge what's related to Angelman syndrome background itself, what's related to the drug, and what's related to maybe the procedure that that you have to use to deliver the drug. Now, we did see a serious adverse event of lower extremity weakness. We definitely saw it in cohorts 1 to 3, the first 5 patients we dosed, and those were patients who were on higher doses. But since then, we've sort of put in some measures where we think the exposure is reduced both in terms of the amount of dose and how long they're exposed and how the exposure is happening. And with the positioning of the bed, keeping it away from the nerve roots, using pre-medications before the dosing. Those are all things we've instituted. And since then, we've seen 3 cases, 2 from expansion cohorts and 1 in cohort 7. But what we want to tell you, like Dr. Berika has also mentioned, is that all of them are doing well. They've been redosed. They've not had a recurrence. So we think that this safety profile in total is very acceptable and consistent. So just to sort of summarize the phase 1 and 2, we have seen consistent developmental gains with sustained improvements, sometimes over 3 years where we have the data available. There have been no additional cases of lower extremity weakness. So the safety profile seems very consistent and acceptable. And with this sort of background with our phase 2 data, we feel that we're really ready, we were ready for phase 3, and that's why we went into phase 3. So phase 3 is our Aspire trial. Absolutely excited to be here to tell you about it. And it's a global study. It's a phase 3 study, and as you heard, and as I mentioned, and you probably are going to be sick of hearing this all the time, but it is a study where you have to have this sort of arm, another arm, including the treatment arm, where you want to study safety and efficacy, and you want to make sure your safety is really real. Your efficacy is really real, because it's not due to, random chance. So you have to have another arm. At least we do need to. Someday, maybe we won't have to. The main eligibility criteria for our phase 3 study is a 4 to 17-year-old individuals with a maternal deletion of UB3A. Should be able to walk independently, not have received any other ASO therapy, and obviously they need to agree to participate in the trials. Our primary outcome measure that we are going to measure is cognition, but we are going to look at all these other domains because it is, again, really important in engagement to look at the whole individual in all the key domains. You can find information in the clinical trials and EU CTIS number. What should you expect in this trial? We'll have a screening and randomization period. After that, individuals will be randomized, meaning it's going to be almost like a flip of a coin. And a crude way of saying it, I know it sounds harsh, but half of our subjects will go into the treatment arm, and half of our subjects will go into what we call the sham control arm. We use the word sham here because sham really here means that although it is the control arm, we are not going to be injecting anything into the lumbar spinal space. What we are doing is we will get some CSF sometimes because it is an untreated CSF that could be used for studies like what FAST and ABOM are doing in terms of natural history and studying natural history for biomarkers. And then we will have, after a year, switch over into sort of the open label period where everybody gets GTX 102. The doses are going to be by intrathecal injection, which is lumbar puncture, as the picture diagram that Dr. Barry Travis showed you. We are going to be testing different specimens to understand all the characteristics of what the body is doing to the drug, what the drug is doing to the body, pharmacodynamics and kinetics. There will be physical exams for safety as well as to look at changes. We will have you complete surveys. Some participants will be monitored with an EEG. And again, this is a global study, and we definitely encourage you to seek out your local sites, and the information for who the local site is and when they're activated will keep populating on the websites for clinical trial and EU CTIS. The one thing I wanted to say again here, thank you again. It's not easy to participate in a clinical trial. So thank you so much. Every step is hard for you. It takes a toll. So the fact that you start and complete a trial is, worthy of applause. I put here on the left side that Ultragenyx has a team that you can consult. These are some of the emails. There are live people behind those emails looking at it, answering them. But it's extremely important to have that relationship with your medical team. Your medical team trumps a lot of the other people. They literally help you decide whether the trial is right for you, the travel is right for you, the duration is right for you, and they are extremely smart, knowledgeable people who have your best interest at heart. So that collaboration with your medical team is very, very important. It's also really important, again, as Dr. Barry Travis said, to have that sort of sense of what does a trial involve, not just for the dosing part, but what does it involve in terms of your responsibilities, in terms of confidentiality, making sure the social media stuff and all of that I won't go into the same detail that Dr. Barry Travis talked to you about, but really important for all of us to be like good stewards. Again, information there. And in summary, I just wanted to say that our phase 1-2 trial showed us a profile that helped us get into our phase 3. Phase 3 has begun. Congratulations. It's global. We are continuously identifying sites. They are being populated as they are getting activated. All our clinical trial participants are going to receive GTX-102 in the long-term extension study, and we are planning that expanded Aurora trial for expanded ages and genotypes, which we plan to open in 2025, so more information will be coming. And with that, thank you. Thank you so much.